All right, so for this lab, we're dealing with 3 selective and differential medias, right? And we're going to define what those are. So we have Mannitol Salt Agar that we commonly abbreviate MSA. So that's the plate I had you find right now, so one we're going to start out with. And then we bought from a company, Columbia CNA, with 5% sheep blood. So these are the blood red because they literally have whole red blood cells in them plates. And they also have 2 antibiotics. That's why it's abbreviated here, CNA, that we're going to talk about. The eosin methylene blue plates, the formula that we use only contains the sugar lactose in it. Unlike in your book, they talk about containing also sucrose. But the formula we use just has lactose in it. So another good reference in your book for blood agar just by itself is exercise 521. Just talks about blood agar without the CNA. So selective medias, these medias are all going to favor the growth of some organisms and inhibit the growth of others. So since each of you guys have a unique organism, depending on the selective ability of these plates, your organism may not have grown because it was inhibited by the selective agents in this media. If yours was selected for, right, it grew, and it was not inhibited by the selective agents. In addition, if it does grow, we can differentiate between different characteristics. So differential media, just like with the differential staining that we've done of gram stain and acid-fast stains where you have gram positive or gram negative, right? We have these different groupings based on their biochemical characteristics, what they can do, right? So when I mention sugar, what's the first thing that should come to your mind when I say there's sugar in this media? What could they do with it? They could ferment it, right? So that's one of the questions that we'll ask a couple of times actually, right, is fermentation. Another one's going to deal with the red blood cells. So the first one we're looking at is the Mannitol Salt Agar, and as I said, all these medias are selective. This one is selective for a particular group of organisms that we're typically looking for in clinical samples. We're looking at Staphylococcus, and the selective ability of this plate is the fact that Staphylococcus tend to be salt tolerant. So what's selective in this media is actually a high salt concentration. So remember the other media we looked at today had salt in it, right? But it wasn't a high concentration. It was an osmo level, right, balanced level of salt. In this case, it's a high level of salt. Oops. So we're going to get ones that can resist this salt are going to grow, right? Ones that can't are not going to grow. Then once we have our growers, we can differentiate between ones that can do something to this plate. So what carbon compound is in the MSA, Mannitol Salt Agar? It's in its name, mannitol, that makes it differential, and it is the mannitol. This is a sugar alcohol actually. So it's a sugar in an alcohol form. That's why it ends in OL. But it is a sugar. So this is how they start out, right? This nice pink color. And because it has a sugar in it that can then be converted into other sugars that can go into glycolysis, right? So that we get pyruvate, we can get fermentation, and we can get acids, alcohols, and gases. Well, we don't have a Durham tube, do we? So we're not trapping any gas, are we? So we don't know if any gas is produced. Would we know if there's any alcohols produced? Alcohol is kind of hard to detect, right? We actually didn't even detect it in the 2,3-butanol test. We detected one of the intermediates, which was acetonin. Acids, on the other hand, pretty easy to detect if you have what in the media? pH indicator. So hence why those plates are a nice, pretty pink color, because they have a pH indicator in it. And it's actually a pH indicator we've already used. Anyone know? Phenol red, right? So like in our phenol red broth, although this one's adjusted to a different pH level, right? So these are more pink. when we start out. But we already know, right, what's going to happen when we get acid production from fermentation of the mannitol. What color is it going to turn? Yellow. So here's a nice pretty streak plate when we had full plates instead of divided plates. And you can see, right, especially where there's a lot of growth, it turned yellow. And where here it's kind of orangey because there isn't a lot of growth, they haven't produced enough acid yet to turn the entire media yellow. Okay. So what is the name of the organism most commonly found in clinical specimens that will grow? It's salt tolerant, so it's probably what genus? Staphylococcus, right? But what is the species? We're looking for Staphylococcus aureus, right? This is the one that will grow and ferment the mannitol and turn the plate yellow. Staphylococcus epidermis, will it grow?
No.
It will, because it's salt tolerant. It's a staphylococci, right? But it will not ferment the mannitol, right? So look at that plate, and you should be able to see the two different staphs that I put on the sample plate for you guys. The staph aureus should be standing out at you because what color is the plate? Yellow. Right? Where the other one you see growth, but did it change the color of the plate? No. Now in the procedure in your book, it tells you to do a Nutrient Agar plate. The only difference here is that, remember, we've been using Nutrient Agar in TSA. It's a control, so you guys can see, right, that it really is the media that's causing these color changes that we're seeing and not the organism, right? It's our negative control, right? So we've been doing it, we're just not doing it on plates, right? And in a clinical lab, they wouldn't necessarily do that because they know what results they're expecting to see. But this is to demonstrate to you guys, right, that it is the media here that's really causing these differences. And that's why we're not seeing growth. So what would be the consequences, though, if someone forgot to add the salt to these plates? The plates would no longer be what? They would no longer be selective, right, for salt-tolerant organisms. So just about anything would grow on the plates, right?
I suppose it's not killing bacteria. Is it not going to kill bacteria?
Right, it's not going to kill anything, right? That's another, it's not going to be selective. You're not going to destroy any organisms because of the salt. So just about anything would grow, so we're no longer being selective. So would this affect the media's specificity or sensitivity? Specificity, right? Because we're no longer selecting, we're no longer being specific, right, as to what we're looking for. Right, so it would be messing with the specificity of this media. So what's in this media? For nitrogen and carbon sources, which we know organisms have to have, right? With the citrate test, we had to give them a carbon, we had to give them a nitrogen, or they wouldn't grow. We were just really evil, right? And only gave them one choice, citrate or ammonia. That was it. In this media contains beef extract, which is both a carbon and a nitrogen source, because this is basically animal cells, right? So it has all the nutrients you can imagine, right? It has sugars and lipids and nucleic acids even, and proteins, right? Sugars have a lot of carbon, proteins have a lot of nitrogen, right? So clearly they've got plenty of food when we put something like beef extract in a media. Peptones are, again, partially digested proteins. So they're a carbon and a nitrogen source. Mannitol, right? Remember I said it's a sugar alcohol. So this is carbon and water. So carbon and H2O, right, is actually what sugars are. They're called carbohydrates because they're carbon and water. Hydrate referring to water is another name for sugars. So just a carbon source, no nitrogen for that one. So, who thinks they have Staphylococcus aureus as their unknown? Look at your mannitol salt plate. Do you have growth? Do you have a yellow color to your media? Nobody, right? Yeah, because guess what? I think the person who has it is the one who's been absent. Who might not be, I guess maybe is not coming back, although I don't know. Okay, I need to check in with her. So, but some of you guys had growth, right? So, do you have staph? Notice I prefaced this with, when we first did it, clinical specimens, right? So is this all stuff that maybe came from a person that we're working with in this lab? No. We actually are using some organisms that are environmental, right? And that could actually be resistant to salt. So again, we can't do just this one test and you would be able to figure out what your organism is, right? So in a clinical lab, it would steer them in the right direction. They most likely are looking at Staphylococcus aureus, right? If it grows and you get that yellow colorization, right? For us, if it grows, what does that mean about your organism? It just means it's salt tolerant, right? High salt concentration tolerant. It doesn't necessarily mean it's a staph. Okay? But you know a little bit more about your organism now, right? So, what do we see? This is a mannitol salt plate, right? I asked someone to do a street plate and they didn't. They just put it in 4 quadrants on the plate. You guys have seen this picture before on a previous quiz. Some of you guys guessed right as to what the person did wrong. But we see growth, right? So what does this tell us about the organism? If it's growing on this plate, what does it tell us about this organism? It's salt tolerant, right? It can grow in the presence of high salt concentration. Could it be staph? It could be. It could be a Staphylococcus, right? But we're just presuming right now. But in fact, I can tell you this was Staphylococcus epidermis that we used to make this actual plate, right? And because it's not Staph aureus, we don't get the color change. So for this plate, notice Staph aureus on this side because it's what? Yellow and growing. You see the growth pattern? Whereas Staph epi on this side of the plate because you see growth but no color change, right? And those are the two that I put on those plates as demos for you guys today. Now, this one we sometimes get with some of our other organisms, right? So notice the organism itself looks kind of yellow, right? But we're looking for color changes in the media because the media is where the pH indicator is. And you can see it's mostly pink over here and a little orangey over here, right? So this one would be somewhat questionable. But I can tell you, in fact, it's not a staph at all. It's just an organism that can grow in the presence of salt, right? And it's not fermenting the mannitol, right? But I don't think we have any ambiguous plates this time, right? We're doing good. Okay. So again, what are the questions that we're asking? We want to know, right, is the organism salt tolerant? And so in this class, we have Staphylococci and some bacilli, although normally bacilli wouldn't be present, right, in clinical specimens, right? They would be more environmental type specimens. So if you have a bacillus, you might be having some growth because it's salt tolerant. So among the survivors, if you have a survivor, then we go one step further and we can ask the question, is it fermenting the sugar mannitol? And again, if it doesn't grow on the plate, we can't ask the second question, can we? Right? Because it would be killed off by the salt. We don't know if it can ferment mannitol. Right? So this is a two-parter. So we inoculated our plates, we put them in the incubator for a couple of days, right? And then we came back and looked at our results. And these I actually left in the incubator. They actually ended up being okay. But usually I'll take them out and put them in the fridge. So the media surrounding the colonies remains red, right, or that pinkish color if there's no fermentation. the media surrounding the colonies turns yellow, right, if you have really good, robust fermentation of that mannitol, right? So we can tell if we have salt-tolerant organisms, right, and if we have salt-tolerant that ferment mannitol, it's pretty indicative that you have Staphylococcus aureus. And unfortunately, we don't have any of those to look at. But again, right, your bacillus, if you have that one, it might also be growing, right, but it's not going to usually ferment the mannitol. So we wouldn't see that color change. So the next one I want you guys to look at is our blood agar plates. And so some of you guys just inoculated these today, right, and so we'll get our results next week. You can tell the blood agar plates because they're blood red, because they literally have red blood cells in them, okay? And this plate right here has an organism that does a particular type of lysis to these red blood cells. There's two different types of lysis, and they do this because they produce exoenzymes called hemolysis, right? And they actually can lyse the red blood cells. What element inside these red blood cells do you think they're after? When I talk about element, I'm talking about carbon, nitrogen, but what's really rich in our red blood cells? Iron, right? Oxygen, yes, but you need that iron to carry the oxygen, and that's what they're after. They need it for their metabolism, right? So our red blood cells are a nice, rich source of it, so a very good target for them. So having this is beneficial for them. Now, in addition to the red blood cells, as I said, all the media we're using is selective. This media is selective for a particular group. What organisms will grow on this media? Gram positive, right? So that's why if you had a gram positive, I gave you this plate today, right? How are we being selective? There's two selective agents in this media. They're abbreviated CNA. They stand for colistin and nalidixic acid. I promise you I won't make you spell them on the final exam. But you do need to be able to identify them, okay? And tell us a little bit about what they do. So they're antibiotics, and so if gram positives are growing, who are these guys inhibiting? Who are these antibiotics inhibiting? Gram negatives. So gram negatives don't grow on these plates. And it's only because these plates have those antibiotics in it that are specific for gram positives, right? Selecting for those and killing gram negatives. So if it grows, right, if it's a gram positive, the antibiotics don't kill it, then we can ask the next question. And the next question deals with what? What is going to change in this media? What's going to be affected? What's going to be lysed? The red blood cells, right? The red blood cells, the whole red blood cells. So again, notice nice bright red color to these plates. They're whole red blood cells embedded in the agar and the other food sources in this media. So what you're going to get is two different types of hemolysis. One that's referred to as beta, with the Greek symbol beta, and this is complete destruction of the red blood cells. So much so that when you hold these plates up to the light, you can see clear through them, right? Because the red blood cells have been completely destroyed, right? So we refer to as this is a clearing, right? A complete destruction. And I always remember beta for bad, right? This is the worst case scenario. Imagine this happening to your red blood cells, right? It'd be horrific. And there are some pathogens out there that do this. So this is the only plate that I have really relatively good, not probably an excellent example of beta, but you can hold it up to the light. I want you guys to pass it around, and I'll explain to you why I don't consider it an excellent example. I think there's some contamination here. There's a little bit of greenish discolorization to this plate, but what I want you guys to look at for this one is the clearing that you see. Okay, so hold it up to the light and see that clearing. This is the beta one, so pass that one around. As that one's going around, on your desktop, there is another red plate, right, that has like a 1 and a 0 written on it. Hold that one up to the light and compare. One side is what we call alpha, that tends to have a greenish discolorization to the lysis. So is it the 1 or the 0 that's alpha? Hold it up and see if you can figure it out.
The one. I would say that.
Yeah. So did you guys see that 1? Definitely has a greenish tinge, definitely seems like the red blood cells have been lysed. The difference with that is alpha is not complete hemolysis like you see with beta. It's what we call incomplete. It's actually poking holes in the red blood cells. And when it does that, it allows oxygen to get in, and it causes this greenish discolorization to that plate. And that's why, you know, that beta one I think has some alpha in it too, because if you looked at it, right, you saw some greenish discolorization too. So I think it might unfortunately be a little bit of a mixture. So some of you guys that have some organisms, most of the characteristics are going to be very similar down to this plate, right? So you're going to really need this plate to figure out which specific organism you have out of the group in the table for, and I don't remember if I added it in here or not. So looking at these two, right, they're alpha Y. They're greenish, right? Do you guys see that? And hopefully you see it even better on the plates that you guys have on your lab bench. This one, what do you think? This is hard to take a picture of. What type of hemolysis do you think we're looking at? This one's beta. This one's beta, right? So complete lysis of the red blood cells. And again, because this is CNA plates, this organism is gram positive. And in fact, if you can read, right, this is Staph aureus, right? And Staph aureus is actually what I thought I was putting in the 0 on those plates yesterday. But clearly not beta, right? There's no hemolysis of the red blood cells associated with the 0, right? And that's what we refer to as gamma hemolysis, which I hate because it sounds like it's lysing red blood cells, right? But it's non-hemolytic. So there are other nutrients in these plates for the organisms, and organisms can grow. So you can have some gram positives, right, that don't damage the red blood cells. So no destruction, right? When we say gamma, we're actually saying no damage to the red blood cells. So this was an interesting plate we got one semester. This is a good streak plate. Notice they did it right? The only problem is they probably breathed on this plate. Because what do you see right here? That's a contaminant, right? That's not their organism. Most of this growth, this other growth, is their organism. Look at one little colony right there, right? And it's what type of hemolysis? Beta. It's definitely beta, right? Staphylococcus aureus, right, is beta hemolytic. There's lots of people who carry this in their noses, right? That's why I said they probably breathed on it, right? Contaminated this plate when they were inoculating it, right? One little bacteria that grew up, right, and caused hemolysis in this one little section on this plate. So it was kind of cool mishap, right? Gave me
Do you think that was Staph aureus?
Oh, I'm almost positive it's Staph aureus, yeah. No doubt. Okay, so again, we didn't do Nutrient Agar plates because remember, we are been doing a slant instead, right? So you guys inoculated your slant. You'll see that you can't see hemolysis, right? Because there's no red blood cells in that media, right? And there's nothing in it to stop them potentially from growing. So you should see nice, good growth, what non-selective medias do. So earlier formulations of the Columbia CNA plates were made using slightly higher concentrations of the two antibiotics, colistin and nalodistic acid, but this was reduced to improve recovery of gram positives. Because think about this, if you have a high concentration of something, even water, y'all, if you drink water and you do not urinate, you will die from water toxicity, right? I kid you not, right? It has happened. Right? And so there was some crazy lady some years ago on a radio station. They did a competition who could drink the most water to win like an Xbox or it was a Wii. Yeah, Wii. She should have went Wii. Because she died. So, anything in too high of a concentration can be toxic. So in this case, right, we're trying to kill off gram negatives, right? And we're not wanting to harm the gram positives. But if the concentration is too high, we could affect the gram positives as well, right? And so what effect would this have on our plates? I mean, what are some possible reasons? I'm all right. I'm skipping ahead. So we've pretty much already answered that, right? Anything in too high of a concentration can affect anything, right? So the concentration is the problem, and the real problem is the colistin, because this one is an antibiotic that affects membrane integrity, right? So who has a membrane? Any cell has a membrane, right? But gram negatives, we know something unique about gram negatives as far as membranes go. How many do they have? 2. So they're more susceptible to colistin because they have those 2 membranes. Now, nalidixic acid interferes with DNA replication. And again, all cells have got to do DNA replication. It's just whether this can get into the cell and actually interfere. And these 2 antibiotics, right, really don't affect the gram positives as negatively as the gram negatives. And the reason for that is cell wall structure, right? So remember, cell membrane here for gram positives, thick peptidoglycan layer. Where with our gram negatives, typically they've got their cell membrane, thin layer peptidoglycan, and they have that additional outer membrane. So they have an additional membrane, right, that's susceptible to damage by the colistin. Right. So colistin, right, this beautiful molecule here, right, is really hydrophilic in some portions and hydrophobic in others. So it's very similar to what we see in membranes, right, with those phospholipids. So it can very easily disrupt the structure. And so again, because of these structural differences, the gram negatives are much more susceptible. But if we put it in too high a concentration, you could affect your gram positives as well. So what are we trying to achieve here? We want to identify and isolate gram positives, and then we want to go one step further once we get the gram positives, like staphylococci and streptococci and enterococci, are typically why they're using this in clinical specimens. Among the survivors, then we can determine if at all, if they lyse the red blood cells, and to what degree. Is it complete beta hemolysis, or is it incomplete alpha hemolysis? And we can determine that, right, by the changes in the lysis, whether it's complete, right, where you clear things around your colonies, or alpha, which is partial and you get that greenish discolorization, or no change at all, right, which is what we call gamma. So if it's gram positive, right, typically we're probably looking at staphylococci, streptococci, or enterococci. Clear halos, we know it's beta hemolytic. And to give you guys an example of that, Streptococcus priogens that causes strep throat is beta hemolytic. And so when I was a kid, back when you go to the doctor and you thought you had strep throat, they literally would swab your throat and rub it on a blood agar plate right there in the doctor's office. Like, I remember this happening. And stick it in the incubator in his lab in his office, right? And then they give you a call the next day and say, go to the pharmacy and pick up antibiotics if it came out positive. And in addition to that, nowadays, you usually don't see them doing that. It gets done behind the scenes. They're going to take a swab, they stick in a special little tube, and they send it off to the lab. But thanks to antibodies, my absolute favorite proteins in the world, which are specific for specific antigens, we have lots of rapid tests nowadays, and you guys are going to perform one in class, the very last lab. We're going to do an ELISA, an enzyme-linked immunosorbate assay, where they're using antibodies for a specific antigen and a color change due to an enzyme in that test. So literally within minutes, they can determine whether you have that antigen in the case of strep. So they swab your throat and they put it into this little test kit, right? And they can tell within minutes if you have a positive result. Now, could you possibly have the infection and not end up with a positive result? Yeah, there's limitations in these tests. Pregnancy tests, same premise, right? There's limitations to them, right? And we're going to talk more about that when we get to that actual testing. So the good news nowadays, though, if they know positive within minutes, you leave the doctor's office and go straight to the pharmacy. You don't wait till the next day, right? So you get faster diagnosis, therefore you get faster what? Treatment, right? And less likely of spread, right? And helping out, and less likely of complications that can happen from strep. Yeah, that's what I'm saying. They do 2 swabs. One, they're going to run the test. The other one, you don't see them put it on the blood agar. They stick it in a tube and they send it off to the lab, and they still do that test. They just do it in the lab now. Right? Because and again, even if the rapid one comes out positive, it's going to be confirmation. Right? It's always good to have confirmation. So ones that are alpha hemolytic is Streptococcus pneumoniae. Right, so again, notice a Streptococcus pneumoniae. For us, our Streptococcus aureus, as we saw in the previous picture, comes out beta, but also some of our bacilli come out beta hemolysis. We have one organism that's supposed to be kind of alpha, doesn't really come out really good, so I'm going to gross you guys out now. How I always know I'm going to get alpha is actually that one I swabbed is from my mouth. Most people have streptococci in their mouth, right? They're the ones that attribute to tooth decay when you feed them sugar, right? So that is actually organisms from my good old mouth that made that nice green one on y'all's plates that I did yesterday. And I have to say, it's not as green as it usually is. Okay, so the last plate, right, is our eosin methylene blue plate. So these are the kind of purplish plates, right? So again, you have samples of these on your desk, and I swabbed 3 different organisms on these plates. And typically, scientists are using these plates looking for a particular group of organisms that we collectively refer to as coliforms. So these are a broad base of bacteria found in our environment, including in the feces of man and warm-blooded mammals. The characteristics that we can definitely observe with this media is that most of them are lactose fermenters. So as I said earlier, we use this media because it has lactose in it, and we use a formula that just has lactose because that's really the question we're asking here, more so than the sucrose, which is just table sugar. Right? That's what sucrose is. It's what you have on your table. So these are gut microbes, hence why they could be found in your feces. They're gram-negative bacilli for the most part. And they produce gas, but again, we're dealing with a plate and no Durham tube, so are we going to see gas? No, right? We're not going to be able to detect gas. But they are going to produce acids from the fermentation of lactose. And when they do that, they interact with the dyes in this media, and they create distinctive color changes. And I have to say, this is one of my favorite ones because E. coli is just absolutely beautiful on this plate.
So it'll fit that brown data, right?
Yeah, so what are we selecting for? Gram negatives, right? Because we just said coliforms are gram negatives, right? So we want gram negatives to grow. But what in the media is inhibiting what's toxic to the most of the gram positives? And notice I said most of the gram positives. Eosin and methylene blue, the 2 dyes, where it gets its name, that's found in this media. And eosin is a red dye, and methylene blue we know is usually blue, right? Under aerobic conditions, which this lab is. So back from preschool, right? When you mix blue and red together, you get purple, right? That purplish color that those plates have. So what group of organisms cannot grow? Gram positives. And again, it probably has to do with the fact that they just have the one membrane, and these dyes are able to get in through it and kill them. where the gram-negatives have the two membranes, and it probably helps them from the penetration of these dyes inward to kill them. Doesn't happen. So, okay, so we've got dyes that kill gram-positive, so if you have a gram-negative, you should have seen growth on this plate, right? And especially if I didn't give you a blood agar plate today, you probably have growth. Really good growth, right? Remember, again, this is not a be-all, end-all. Some gram-positives can grow, right? Although it's rare. Then the next question is, if it grows, what is it fermenting? Well, I already said lactose, right? That's what's going to give us the differential. So typically your book says lactose and sucrose, but the formula we use in lab, as I said, is just lactose. So they only have one sugar choice. So E. coli is a fecal coliform, right? So it's in this classification. It's a gram-negative bacillus. It ferments lactose and produces gas, and it's a really good fermenter. So what color does it appear on these plates? It's going to appear black and or metallic green growth, right? Because the interaction of the acids with these dyes. It's really pretty. So you should very easily be able to find it on that plate that I had for you guys, right? If you flip it over and actually look at the side where you see them growing, don't look at the bottom of the plate, look through the lid. Or lift up the lid if you need to, if there's condensation. You guys see that nice, pretty metallic green on that plate?
You remember using these plates before?
Yeah, I was about to get to that.
Oh, sorry.
So we used these plates once before when we did our counting activity. And do you guys remember that we had E. coli? And so you've seen this color before, right? You just didn't know what you were looking at and why, right? So our plates, the couple plates that we had, and that was kind of a confirmation too that we didn't really have contamination, that we actually had E. coli growing. And again, a lot of times when they do these, they would do that because they're specifically maybe looking to count just these guys as opposed to any other bacteria. So an experiment I used to do in the past is I actually used to take hamburger meat and grind it up in the blender. And hamburger meat can sometimes contain E. coli as well as other fecal coliforms. I won't continue to gross you guys out, but you know, there's an English professor here apparently that is really educating her students about the foods we eat and where they come from. But yeah, sometimes it can be contaminated with organisms from the gut of the cow. And this is why, right, it's very, very important to cook your hamburger meat all the way through, right? Don't be doing a medium well burger unless it's a steak you ground up yourself.
That's what my brother does.
Yeah, I have a friend that, like, I kid you not, every time, I'm like, why do you even order hamburgers? I was like, because, like, we tell the waiter, we're like, cut the hamburger in half before you bring it out. Make sure there isn't any pink, red, you know, that it's brown, that it's like dead, right? Before you bring it to her, because she's just going to keep sending it back until it looks like that, right? And so, like, the waiter's like, you know, and I'm like, dude, seriously, don't bring it out unless you've cut it in half and it's brown. Because she's just going to send it back. Because when she was a kid once, she got a hamburger at a restaurant and she got really sick from E. coli contamination. Like, seriously sick. So she doesn't mess around. But again, I'm like, why do you even eat burgers then? I probably wouldn't even eat burgers if it was that bad. My sister got food poisoning from shrimp from a Chinese restaurant, and she hasn't eaten any since then. She's like, I'm done. I'm done. No, thank you. So, yeah. And the bad news with, you know, E. coli, there are some pathogenic strains. But if you have E. coli in your hamburger meat, it doesn't necessarily mean it's a pathogenic strain. It not necessarily mean you're going to get sick. But the fact that it's there, other organisms that can come from that same area from the feces could also be present, right? So this is what we kind of call an indicator. Because, I don't remember if I have it in here or not, Shigella, right? Which is a really mean one, right, for food poisoning. That one's not a lactose fermenter. It's a gram-negative, but it's not a lactose fermenter. And in fact, most of the really pathogenic ones are not lactose fermenters, but they usually are gram-negatives and will grow on this media. If the organism ferments the lactose and produces a small amount of acid, right, so E. coli is a really strong acid producer. There's other ones that have different levels. What kind of colors should we see? We should see pinkish to purplish colors. And again, this is a sample that I did with a class from hamburger meat. This was growth that we got from a sample from hamburger meat. Raw hamburger meat, uncooked. So, and I'm going to do this again this summer with my high school students. It's going to be fun. Gross them out. So what's the purpose again of the Nutrient Agar? Just a negative control, right? So you guys can see, right, help validate. And in a clinical situation, they wouldn't do it. Okay, so dipotassium, phosphate is a buffer that's added to the EMBs that help adjust the pH to the proper starting level. So remember, what are we producing when we ferment? Acids. Does that affect the pH?
Yes.
Yes, right? And a lot of times, a lot of this media that we utilize for fermentation with acid production, we have buffers. And buffers are always kind of equated to the bumper on your car. It helps cushion the blow, right, when you're in an accident, if somebody bumps you, right? If someone slams into you, that bumper does you no good, right? And so the same thing here, over time the buffer will get overwhelmed by the acid production. But the purpose, as they're saying here, is to make sure that we're starting at that level that we want to start at. So what would be the consequence if we started at a raised pH of 7.8, right, above what it should be? It would be too much towards the alkaline, right? 7 is neutral, 7.8, we're kind of pushing a little alkaline, which means what does the organism have to do to create those color changes? Produce more acid. So what we're creating a problem here, right? So it's supposed to be around neutral or a little bit less than neutral. If we go up even just this small amount, it'd be more difficult to determine if they actually produce those acids. We're going to be less likely to get those color changes, right? So, for that consequence we just talked about, would that be messing with the sensitivity or specificity of this test? This would be a sensitivity, right? Difficulty reading the results, right? We're not changing the specificity. We're not taking the sugar out or putting a different sugar in, right? We're definitely testing for lactose fermentation here. We just may have some difficulty detecting it, right, if our pH levels aren't where they need to be at the start. It'll make it harder for the organism to create that color change that we're looking for. So again, you become aware of the diversity and the abundance of bacteria, right, in our environment and that we're exposed to. With this in mind, right, if you see a metallic green sheen, is it truly a coliform bacteria? Is it definitely E. coli? No. It could be some other organism, right, gram-negative, right, because these are selected for gram-negative. That's a really strong lactose or sucrose fermenter if you're using that formula of the media. So again, although with these tests, and especially in our lab, right, when in Antarctica, if it looks like a penguin, it probably is a penguin. And so in the case of our lab, if it looks like E. coli, it probably is E. coli, right? And unfortunately, our one student who has E. coli, it didn't come out as it's supposed to look. So I think we got some contamination there. So we'll have to redo that one. So with these two plates, right, notice how there's really none of those colors that we talked about, right? We're not seeing the black, we're not seeing the metallic green, we're not really seeing purple or pink, right? They're kind of blending in with the plate, right? We're not seeing a distinctive colorization. But they are growing, right? You can see the growth on these plates. So are they gram positive or gram negative? They're gram negative, and why are they gram negative? Why don't we see gram positives? What happened to them? What's in this media that's going to inhibit gram-positives from growing? The dyes, eosin and methylene blue, right? So the dyes have killed off the gram-positives. Are these organisms fermenting lactose?
Yes.
What are color changes that indicate fermentation? The metallic green, do you see that? Do you see black? No. Do you see like a bright pink or a bright purple? No. These are not fermenting. No. You don't see the pink, you don't see the purple, you don't see the black, you don't see the metallic green. These are non-lactose fermenters. Gram-negative, non-lactose fermenters. Are these organisms gram-positive or gram-negative? Well, they're growing, right? So they're gram-negative, right? Again, because the gram-positives are killed off by the dyes. Do we see any color changes?
No.
Look closely at this one. It's kind of pinkish. I know the picture doesn't do it justice, but it's a little pinkish purple in color. And we have a couple that do a little bit of fermentation, but we really have to look at those closely, and it's hard to see in pictures. So our last one, right? So why did we do this? So we want to identify gram-negatives, right? So this is a confirmatory for those of you guys that have gram-negatives. Especially we're looking for coliforms that can ferment lactose. So if it grows, the next step we're looking for those fermentation products, those colors. We just simply inoculated the plates, right, with our organisms. If they're colorless, there's no fermentation. If they're pink, there's a little bit of fermentation. Purple, black, or sometimes that metallic green, really strong fermenter of the lactose, right? Indicative usually of Escherichia coli. Other organisms, Enterobacter, Klebsiella species, these will produce their gram-negatives, and they'll have some lactose fermentation as well. So let's see.